Good morning or good evening as I record this talk. Thank you very much for inviting me to share with you the results of some of the recent work from my group. My name is Gosia Trynka. I lead the research group here at the Wellcome Sanger Institute, and I'm also the Director of Experimental Sciences of Open Targets. And today I'll share with you some of our approaches that we use to interpret GWAS variants associated to immune diseases using specifically chromatin marks for that. So common complex immune diseases, which are the predominant focus in my group, collectively affect about 10% of general public. And although they have a broad range of manifestation where they can affect specifically different organs in our body or have more systemic nature, one thing that they have in common is dysregulated impaired immune system. And additionally, immune diseases have been at the forefront of genome-wide association studies. And as a result, we currently have thousands of associated risk alleles that give us an anchor to disease biology. However, translation of this GWAS associations to function has been somewhat challenging. And one of the reasons why it's been challenging, particularly in immune diseases, is the fact that often it is unknown which of the cell types is the disease-relevant cell type. And in immune diseases, there's really a whole range of different immune cell types and different states that can be involved in disease pathobiology. And because majority of GWAS variants are non-coding, they are implicated to play a role in gene expression regulation, which can often be cell type specific. So really one of the critical first steps in efforts to interpret GWAS variants is identifying which cell types are disease relevant. And once we have those relevant disease cell types, then we can focus on our efforts more on more functional studies to refine those GWAS signals to disease causal genes, disease regulatory causal variants, and more broadly to cellular functions that might be impaired by the disease-associated variants. Now, there are a number of different approaches that one can use in order to address these challenges and interpret GWAS signals. And we have outlined together with a very talented PhD student from my group, Eddie Gomez, we've outlined some of those approaches in a review that was published earlier in the year in Frontiers in Genetics. But specifically, one approach that I'll focus on in here includes integration of GWAS variants with chromatin activities or chromatin marks. And this approach, in a nutshell, has a very simple idea behind it. It really relies on overlapping GWAS variants with these peaks from chromatin assays. And the approach does it collectively across multiple loci that are associated to a trait. So in this simplistic example, we can see that the variants at these two independent loci are overlapping peaks that are specifically active in bone tissue in this case. So when we do that across a number of different loci, we can then compute this enrichment score. And that tells us that certain variants are enriched in one tissue over the others. And now, once we have this identified tissue or cell type that can be disease specific, we can go on to more detailed subsequent functional studies. And one of the approaches can be to recruit larger numbers of individuals and perform functional genomic assays that then can be correlated with genotypes. So in this case, such approach that essentially is a quantitative trait locus mapping, can be applied both to chromatin marks, where activities or reads within individual regions, such as this one, can be correlated with genotypes to then assess if there are effects of different alleles on activity of chromatin, on chromatin activity. Or more broadly applied, this approach is for gene expression, in which case we can identify if variants correlate with gene expression levels. And once we have such maps of QTLs, then we can ask a question if these signals at individual QTL loci share a pattern of association with GWAS loci. And in instances where indeed these patterns are shared, we can call such events as colocalizing events. And essentially what that tells us is that there are shared causal variants that are driving both signals for the disease association as well as for the molecular trait, which can be, for example, chromatin activity or gene expression. So with these two approaches in mind, I'll show you how we've been gaining insights into the dysregulation of immune system in immune diseases by the variants associated with different immune traits. So quite a while ago already, we've applied the Snip enrichment analysis to a range of different immune traits. And what we observed was that disease-associated variants were enriched in chromatin marks that were specifically active in CD4 T cells. And interestingly, what we observed that regulatory T cells were coming up as enriched across a whole range of different immune diseases. Now, regulatory T cells are an infrequent cell type. They are a subset of CD4 T cells. And although they are infrequent, they actually play a really critical role in regulating the immune system. And very simply, to illustrate you what's the function of regulatory T cells, essentially, CD4 T cells play a critical role in mediating the immune response. So when they encounter an antigen-presenting cell that presents an antigen on its surface and they recognize the antigen, the conventional T cells undergo activation and proliferation and then interact with a range of different immune cell types to really direct how the immune system should respond to a pathogen that's present in our body. So clearly a really important process. However, this process also needs a braking mechanism, and this is really where regulatory T cells come in. Their function is to suppress this T cell activation and reduce the inflammation and the immune response. Because TRex are an infrequent cell type, we noticed that there weren't any publicly available data set that would allow us to follow up this SNP enrichment signal in TRex more substantially. So what we did was to recruit over 100 healthy individuals and then isolate TRex from those individuals. And following TRex isolation, we perform a number of different functional genomic assays. So we profiled a gene expression as well as we profiled chromatin activity using 3 different chromatin marks. So we've used ATAC-seq to map chromatin accessibility regions. We used H3K27 acetylation, which tells us about active enhancers and promoters. And then we've used H3K24, H3K4ME3, which tells about gene promoters. And as a result, we were able to perform QTL analysis for each one of those individual assays. And as you can see, for almost 30% of genes expressed in TRex, we were able to see that there was a genetic effect on gene expression. On the other hand, we were able to map far more chromatin regulatory regions using the 3 other chromatin marks. And as you'll see, the number of these regulatory regions that are under genetic control was different between the different assays. And in particular, we saw that acetylation QTLs, which reflect H3K27 acetylation activity, were more abundant across the 3 assays. So with this map of eQTLs and chromatin QTLs, we then performed the colocalization analysis. And we observed that across a range of different immune diseases, we were able to identify tens of events where we observed colocalization between GWAS-associated signals and QTLs in our data set. The different colors here represent different assays, and what you will quickly appreciate is that the majority of colocalizing events occur between eQTLs, between gene expression and H3K27 acetylation QTLs. And now, very briefly, how we can use this data set. So essentially, this really rich resource allows us to now nominate what can be potential causal genes, because we have this colocalization events between gene expression eQTLs and GWAS-associated loci. So we're able to nominate what are the likely causal genes at this loci, but also with chromatin QTLs, what we can do is ask a question if the associated variants are overlapping chromatin peaks. So here's an example of one such locus where we can see that an LD block of 14 associated variants that pose risk for IBD. Of those 14 variants, 5 overlap a chromatin QTL peak. This is an acetylation peak, which gives us an idea that probably of the whole block of 14 variants, 5 that are overlapping this chromatin peak are most likely functional variants. So with this approach of overlapping variants with chromatin peaks, you can see that we were able to reduce the numbers of associated variants at each individual locus from hundreds often down to tens of variants that we now prioritize as likely causal variants. And now that makes it, that list is now of variants is now reduced and allows us to design experiments, functional follow-up experiments where we can dissect the effects of those variants with in more detail. One thing that was quite puzzling to us was the fact that we observe for a large number, actually for majority of the colocalization events where we observed colocalization between acetylation QTLs, we were unable to link those acetylation QTL events to downstream gene expression events. And so this is really interesting, and I'll show you one example where we put a little bit more efforts into understanding what exactly is going on behind this colocalization with an enhancer eQTL. So again, this is the example I showed you earlier. This is an enhancer that is an intergenic enhancer. And the eQTL is really strongly colocalizing with variants associated with IBD risk. This locus is not only associated with IBD, it is also associated with other diseases such as psoriasis, IBD, asthma, and type 1 diabetes. It turned out that our collaborators, Rahul Raychaudhuri and his team at Babraham Institute, were also interested in this locus. And they, what they've done is they deleted this enhancer in mice, and they observed that upon deletion of the enhancer, there was differential gene expression that was affecting this LRRC32 gene. And this was specifically focusing on TRex. This LRRC32 gene encodes a GARP protein. And now mice that lack this enhancer actually were more susceptible to develop induced colitis, so very much reflecting the phenotype of IBD-associated variants. Additionally, what they were able to show is that not only those animals were more susceptible to developing IBD, also the TRex had an impaired suppressive capacity. So in experiments where TRex were supposed to suppress the inflammation, actually that wasn't the case, and they were able to see that the animals were actually not recovering, and they were progressing onto the disease. Interestingly, this effect in vitro studies was really shown to be specific to activation of TRex. So they observed that this impaired expression of a GARB was manifested when when TRex were activated with IL-2. And now that brings me to the other point that I would like to make today, which is the importance not only of identifying what are the disease critical cell types, but also identifying if there are specific cellular contexts that are most relevant to the disease. And again, focusing on CD4 T cells, such context can be, for example, provided by different expression of or different presence of cytokines in the environment. So as I mentioned, CD4 T cells play a critical role in directing the response of the immune system. And one way through which this is achieved is when CD4 T cells are activated, the presence of the cytokines in the environment helps T cells to acquire different phenotypes. And then depending on the phenotypes that they acquire, they go on to communicate with other immune cells in specific way. So we devised a project that was funded by the Open Targets and really has been driven by a fantastic, very talented trainees in the group, Blage, who's a postdoc, and Eddie, who's a PhD student, together with Debbie. This project, the goal of this project was to isolate CD4 T cells and monocytes, which are an antigen-presenting cell type, isolate those cell types and stimulate them in the presence of different cytokines, which you can see in here. And then upon activation, we profiled chromatin activity in these different cell types, and we've done it at 2 different time points, which were different between T cells and macrophages. And collectively with this approach, we generated a resource of 55 different cell states in immune cell types, and we used 2 different chromatin marks to profile chromatin activity. What we observed, first of all, was that upon stimulation, we observed an induction of large number of peaks. However, what we also observed was that the majority of those peaks were actually shared between the different cell states. So only really a small proportion of cells, a small proportion of peaks was cell state specific. On the other hand, when we looked at how variable those peaks were between the different cell states, we actually observed that there was quite high variability. And so that was interesting, because what we wanted to do with this map was to apply this Snip enrichment approach that I told you about at the beginning, which relies on this principle that the variants are overlapping peaks present in one cell type and the peaks are absent from the other cell types. Now, that wasn't the case in our data set. What we were dealing with more was this scenario where we have variants that are overlapping peaks, and those peaks have different activity across the different cell states. So Blage thought that this was calling for developing a new method, which is exactly what we did. We developed a method that's called CHEERS. And I'm not going to go into details of how this method works. I do invite you to read a paper where we provide details on the method, or I'm happy to discuss it after the talk. But essentially, this Snip enrichment method now takes into account the activity of the different peaks. So when we applied it to our data set, what we observed was that the method was sensitive enough to identify that there were specific conditions where we observe enrichment of variants in individual cell states, such as, for example, in IBD and celiac disease, we observe strong enrichment in Th1 phenotype, which is a cell type that has actually been shown to play a role in pathobiology of those diseases. But more broadly, what was striking to us was the fact that actually across a whole range of different immune diseases, we didn't see that there were individual cell states that were particularly enriched for disease-associated variants, but more we observed that it was these variants were pointing towards this regulation of early activation of memory T cells, which are colored here in a light purple color. And when here is just to illustrate you an example of how this, what SNP overlaps are driving this enrichment. You can see these are allergy associated variants, and they are overlapping peaks that are really specific to CD4 memory T cells, and the region is quite flat in naive T cells, and especially the peaks are upregulated in early activation of memory T cells, and the activity of this region is much reduced after 5 days of activation. And what we've also done is we took this loci and took all the genes nearby these peaks and asked a question: what pathways are enriched for? And we observed that indeed a lot of the pathways were pointing towards T cell activation and lymphocyte proliferation and so on. So I'll leave it here because in my next talk I will tell you a little bit more how we're following up these signals. But in summary, what I would like you to take home from this talk is that SNP enrichment methods with chromatin marks can point towards disease-relevant cell types and cell states, and identifying those cell types and cell states is really critical for functional follow-up studies. It looks like disease variants can often regulate gene expression in a cell state specific manner. And finally, for immune diseases, we're now building evidence that disease variants are disrupting the T cell activation circuitry. And with that, I'd like to thank the team very much, especially people who featured the work that I presented here are Blagoje Šoškić, Eddie Canogames, Daphne Glinos, and Lara Bosin Castillo. And also big thank you to our collaborators, Raul Rai Chowdhury. And with that, I'm happy to take any questions.